Sorry for being a little bit late. I had trouble getting Zoom up and running today. You should also note that my clock probably is not correct. I need to take it into Clark College to get the time correct on this clock. Before we begin, are there any questions? All right, let me make a couple of announcements and then we'll begin. First, I might be called up for jury duty next week, in which case, if I am selected for jury duty, I will not be able to teach this class at the normal time. I don't know, and I probably won't know until Monday, whether I'll be selected for jury or not. So just be warned that next Tuesday, I'll send out an announcement, but this class may not be running if I'm selected for jury duty, in which case I'll post something online and then you can see it online. Or you could probably go to my 6:30 lab because I think that one's late enough that I can get home in time to make that lab. I'm not sure about a 5 o'clock lecture because I'm not sure what the times of the jury duty will be and how long it'll take me to get home. But um, like I said, I'll make an announcement, and if I'm selected, it'll be a disruption to her class, and sorry about that. There's not much I can do about it. I've requested that I be not selected for jury because it'll interfere with her class, and there's no one who can substitute as a teacher either, because the main person who would do it is now department chairman, and he can't do it now. He's way too busy. So we will might be going online if I'm selected for jury duty. Any questions about any of that? Let's see, I don't need this right now. Probably close that down because it'll make noise if I get started on the class. Um, One other announcement is I've learned that if you check out a computer from Clark College, I'm not sure if this is from the computer hub place or from the library. It might be both places. They've already got Respondus Lockdown Browser and Respondus Monitor on those computers. And then to get those programs to work, you have to use the programs that they downloaded. If you download the program on the computer, it won't work. So if you have checked out a computer from Clark College, you should check to look and see if Respondus Lockdown Browser and Respondus Monitor are already on your computer, and then just use those. Because if you download it yourself, it won't work. Any questions about any of that? And that's something new. I don't think they had those on the computers earlier. Maybe they have done it for a few terms and I just don't know about it, but to me it's new. Any questions about anything before we begin? If not, let me go to the syllabus, show you what we're doing. Can somebody confirm you guys can hear me?
I can hear you.
Thank you. All right. Excuse me. Today is Lab 1. We're going to be talking about the ubiquity of microorganisms. Remember that this lab must be turned in by 8 a.m. Friday morning so that I can find out who is attending class and who is not. And anyone who doesn't have Lab 00 or Lab 01 turned in will be dropped from the class. Let me remind you, you have to take the online plagiarism quiz by this Saturday, 11:59 p.m. You must achieve 100% on the plagiarism quiz to get full credit on the infectious disease project. You may take the online plagiarism quiz up to 7 times. They'll be the same questions, although the position of the answers will shift. You should also read the infectious disease research project. We will begin that project next Tuesday. Let me show you where that is. Under Lab 2, Week 2 Lab, Infectious Disease Research Project. I'll be talking about that next Tuesday, and I will then open it up for students to submit which infectious disease they would like to select. I will probably, because this is a very large class, there's going to be 2 labs partaking. I will probably narrow it down. Like somebody's, well, no, they haven't, but somebody's requested AIDS, and I told them you have to submit it on Tuesday. Tuesday morning will be fine. But uh, AIDS and HIV, and I told them you have to break that one down into HIV-1 or HIV-2 and only talk about the topic you've selected. And in talking about the other, you can only talk about it in passing, meaning briefly mention it in a sentence. Um, but so most of you, I'll try and narrow it down so that we have more people actually getting their first choice selection. Because with 40 people in the class, it would be very difficult to come up with 40 different diseases. Any question about any of that? Anyways, read this by Tuesday. Do not select a topic until Tuesday. Okay. So don't email a topic until Tuesday because I'm not going to record it. I'll just tell you, you have to email it to me on Tuesday. All right. If there's no questions, let's talk about Module 1, Lab Module 1, Ubiquity of Organisms, and you'll get your questions and submit your answers in worksheet here, which, by the way, I don't have the button to submit it. I guess I can show you that. You'll have a button over here to submit. I've got a button to grade it, okay? And if it doesn't show on the right, it'll be showing down here. Questions about any of that? If not, let me go ahead and go to the lab module. Lab Module 1, Ubiquity of Microorganisms. To each lab, there will have a bit of reading to do before you work on the lab. So for this lab, you should read pages 156, 157 to 162 of the college's version of the textbook, meaning the third version made for Clark College. And that'll be talking about culture media. You should also read about obtaining peer cultures. That's only a page, page 162, 163. There are some video clips throughout this lab module. You should watch those. The learning objectives for this lab is upon successful completion of this lab, students should be able to know that microorganisms are essentially almost everywhere on the planet. You should describe the purpose of culture media, describe the terms colony abundance and colony morphology, and then you should define the following terms. What's inoculation? Ubiquity of microorganisms? What's something that's sterile? What's an isolation? What is incubation? What is in vitro, like in vitro growth? What are culture media? We have different types of culture media. They can be a broth, that's a liquid culture. It can be a solid, that's usually an agar dish or an agar tube. And when we're talking about an agar tube, we can have 2 types, a slant and a deep. What is a fastidious organism? I suppose I should say what's a non-fastidious organism. what is non-selective media, and what is something that's ubiquitous, and what's a colony. Any questions about any of that? All right. Microorganisms are organisms that are too small to be seen with the naked eye. This does include, at least with microbes, what are viruses and prions, even though they aren't alive, even though they aren't really organisms because they don't have a cell, but we do lump them together as a microbe. We will also include helminths, worms, in our studies, even though the adults are not microscopic. However, their infective stages usually are microscopic. Any question about any of that? So microbiology does study parasitic helminths or parasitic worms, even though the adults are not microscopic. Everything else that microbiologists study are microbes, meaning something you need a microscope to see. The phrase ubiquity of microorganisms refers to the concept that microorganisms are essentially everywhere. That's not entirely true. You don't find them become a couple of places on the planet, and we'll talk about that. But microorganisms are almost everywhere. When we're talking about something that's sterile, like when I hand you a sterile tube, if you were to come into a real microbiology lab, that would mean that it's free of living cells. Like hopefully if you go undergo surgery in the hospital, it's done sterilely. In this lab, we will investigate the abundance and diversity of microorganisms in common environments. We'll also learn about basic lab protocols. A culture media, also called a growth medium, is a sterile laboratory preparation that contains a variety of nutrients that are required for growing microorganisms in vitro, meaning in a lab. A culture media may be liquid. We call these broths. They're usually used in a test tube. You can add agar or some other solidifying agent to the broth to change it from a liquid into a solid. We usually add agar, but we could add something else like starch or gelatin. Here are two videos, or I guess one video for you to watch uh, on how to prepare pour plates. Note how the culture media needs to be sterilized prior to its use. And in the lab, how we get that is we put it in the autoclave and it will sterilize the media. Some important questions to consider with preparing pour plates is why must a culture medium be sterile prior to being used? There we have a liquid broth in a tube, and it's usually put in a tube because it can easily spill out of a plate. And then this, that, and these are solid medias. This is what we call a slant, where the agar surface or the media solid surface is actually in a slant. This is called a deep, where you just pour the agar into the tube and then let it solidify. So that's an agar deep. And then these are sterile petri dishes, which usually have solid media in them because if you had liquid media, it'd be kind of easy for it to spill out and contaminate the dish. All right, today we're only going to talk about one type of media. It's a general purpose media. It's non-selective in function, meaning it does not select for specific types of cells to grow. General purpose media are usually complex in composition and they support the growth of most non-fastidious organisms. Something that's non-fastidious can grow in a general purpose media. meaning non-fastidious. You don't need to add special reagents to the media to get the cells to grow. Some examples of general purpose media that we use all the time in microbiology include, I'm going to start with number 3 first, nutrient agar, a very simple media containing mainly ground up beef extract, but also sodium chloride and water. And usually we add agar to it. Well, we call it nutrient agar. A tryptic soy agar, another very common general purpose media. It uses trypsin, which is a protein they derive from soybean. So we call it tryptic soy agar because the main ingredient comes from soybeans. And then nutrient agar with yeast extract. It's nutrient agar with extracts of yeast added, so it's a little richer than nutrient agar. But all three of these, that's abbreviated NA for nutrient agar with yeast extract, nutrient agar abbreviated NA, and tryptic soy agar abbreviated TSA. Any questions about any of that? There are different types of media, but we're not going to talk about them in this lab. We'll talk about them in a future lab. When you have something in the microbiology lab with the culture in it, it is vital that all Petri plates and tubes be correctly labeled prior to their use. The following 5 items m- must be included on the tube. One of them's optional, but you must have your name on the tube or dish. You must have the date. So there's somebody's name, there's the date. You must have the type of media on it. Yeah, let me see if I can scroll down there. TSA is the type of media. The inoculum, what you're going to be growing on the tube or the dish, and this would be E. coli growing here. You can put just like exposure to the air, in which case you bright air exposure if you don't know what you're growing. And then the incubation temperature is optional. You can write that, you don't have to, but this was growing at 37 degrees C. For a tube, usually you write on a tape and then attach the tape to the tube. On the dish, you actually write on the bottom of the dish, usually around the edge or on the edge, because if you were to write right here, it'll interfere with your seeing the culture growing on the tube, on the plate here. So usually around the edge of the tube or on the edge itself is actually the best place to write it. Inoculation is the placement or transfer of microbial specimens into a sterile culture medium. Incubation is the placement of the inoculated culture media in an appropriate physical environment for growing the microbes for a specific period of time. Usually we incubate things in what we call an incubator, but you could incubate it like at room temperature. Um, but usually most things will grow in an incubator. We have different types of incubator. There are complex ones that include uh, providing a specific amount of oxygen, providing a specific amount of carbon dioxide. They all can control the temperature. They generally all can control the humidity, and they all allow you to grow it for a specific time. In the lab, we only have simple incubators, and they do not allow you to select the oxygen exposure or the carbon dioxide exposure. Only you can select air. Let me show you a complex one. This is a complex incubator that has a bunch of dials down here for controlling the amount of oxygen and the amount of CO2. Usually you have a little dish, actually a fairly big dish, that you put in the bottom of the incubator and you put water in that dish. That allows the incubator to be humid, meaning the water will evaporate from the dish, adding humidity into the incubator. That's important because if you don't add water, the dish will dry out quicker. After a few days, you will only be able to get growth for a few days. If you add water and make it a humid environment, the dish will last, a petri dish will last at least a week, sometimes as long as 2 weeks, but at least a week. Like I said, we have simpler incubators and they can only control the time that you can grow it for, the temperature you can grow it at, and you can put the dish of water in there so it supplies the humidity for the incubator. Any question about any of that? So you should note that during incubation of agar plates, the water can come out of the agar. Let me make this an example. Let's pretend this is a plate and we have agar down here. The water can come out of the agar and then condense on the lid of the plate, forming a droplet. And then the droplet can drop down on the plate, which would spread your colonies and your growth around on the plate, which we would not want. It does not necessarily contaminate the plate. However, if you were to take the lid off and then move it, the drop of water could come down the side of the lid and then drop into the plate. And that likely would contaminate the plate because the side, especially the bottom side, is not sterile. The top of the lid would be sterile though. So it would increase your risk of contamination, but it may not contaminate the plate. But it would disrupt your growth. We don't want that. So what we do is when we put a dish in the incubator, if it's a solid dish, you put it in, flip it upside down, and then you put it in the incubator upside down. Now, if water comes out of the agar, it would condense at the top, and so what, even if a drop were to form, it would drop on the lid. We don't have anything growing on the lid, but actually the agar will wick the water so that the drop would not ever form on the agar. So we put everything in the incubator. If it's a solid dish, we put it in the incubator upside down to prevent the drops of water from dropping in the dish and then spreading your growth around. Any question about any of that? If you see a colony on the agar plate, it will have tens to hundreds of millions of cells. To be visible on a plate, you have to have a colony with at least 1 million cells. Anything smaller would not be visible, at least to the human eye. And sometimes each colony could form from a single cell. And what happens is that single cell will divide into 2 cells, divide into 4 cells, divide into 8, 16, etc., until you get around 1 million cells. And then you can see the colony. Now realize when you do grow colonies on a dish, not all of them will be formed from a single cell. Almost about a third of them will be formed from more than one cell, either 2 cells, a few cells, or a clump of cells. And that may explain why some of the colonies are larger than the others, and that is they were formed from more than one cell. But the point is, you could have a colony containing millions of cells, each of them derived from one cell. There's a little video you can watch on binary fission, how one cell can divide into two and become mini cells. It's a very short video. And there we have an agar plate having growth on it. And you can note that you'll have different colonies that you can distinguish as different species. Like that's definitely a different species than that one, which is a different species, both of those two, from this one and that one, and probably different from that one as well, because that's a different color and a different colony shape. And that would be a different colony too, because a different um, color to the colony. Any question about any of that? So, when you have a colony on a plate, each colony will be at least 1 million cells, otherwise it won't be visible. Sometimes the colony can be formed by one cell, like that one maybe, small. Other times they may be formed by a clump of cells, like this one. It's very large, or maybe that one. And like I said, you can see different species by their appearance. You can also determine by their appearance whether it's bacterial, meaning the colony edge is sharp, well defined, or whether it's a fungi, because the colony edge is not so sharp. Let me blow that up a little. It looks kind of hairy and fuzzy. And that one too is a mold colony, although that one is a little sharper, it's still fuzzy, and at least in the center. And usually mold colonies are larger than bacterial colonies, but that one's not the best characteristic to determine. Usually by appearance, you can determine what is a fungal colony and what's a bacterial colony. Now, not all bacterial colonies are well defined and distinct, but most of them are. And then not all fungal colonies are hairy and fuzzy, but most of them are. Colonies will not be visible in a liquid culture because the cells can all mix together in the broth and you cannot see an individual colony. You can only see turbidity in the tube, which is a bunch of cells growing in this tube, but you cannot see individual colonies. And so you would not see different species in a liquid broth, but you could see different species on a solid plate. When we see different colonies on the plate, you can see obvious visible colony morphological variations from different colony species. We have different colony characteristics. You can get the shape of the colony, and you should know circular colony, an irregular colony, a spindle colony. I would not ask you to distinguish between a rhizoid colony and a filamentous colony, but the point is there's different colony shapes. There's different colony margin. The margin is the edge of the colony, and so if we have a round circular colony, the margin is entire. But the margin could be undulate, lobate, or curled. I would never ask you to distinguish is the margin undulate or lobate or curled because it's too difficult to distinguish. And then I could ask you, is it rhizoid or filamentous? But I would never ask you, is it rhizoid or is it filamentous? Because these two are too similar. But you should be able to say the colony, by looking at it, is it an entire margin or is it undulate, lobate, or curled margin? Or is it rhizoid or filamentous margin? You should be able to distinguish the elevation, but you'd have to really look at it, the petri dish, directly, and you have to take off the lid and then look at the colony edge. Is the colony flat? Is it raised? Is it convex or pulvinate? Distinguishing those two would be too difficult. Or is it umbonate? So I could ask you that. Although when we look at colonies in a picture, it would be very difficult to determine, is that colony flat or is it convex? Like these are probably convex, but it's hard to distinguish. It could be a mesa, it could be flat, but we do have different, a mesa is raised, sorry. We do have flat colonies raised, convex and umbonate, but it's very hard to distinguish that in a photograph. You have to look directly at the plate, take off the lid, and then look at the side of the colony. You can determine the size of the colony. Let me see if I got a pencil here. Here we go. Punctiform is very small. That would be a colony that would be less than the size of a pencil. We're talking about a little dull pencil, meaning not terribly real finely pointed, but the average pencil point. If it's smaller than that, it's punctiform. If it's about the size of this pencil, it's a small colony. If it's between the point of the pencil and the eraser, we call it a moderate size colony. And if it's larger than the eraser, that would be a large colony. Any question about any of that? You can look at the colonies and determine whether they're smooth or they're rough. Students often have a trouble distinguishing that. But as you can see here, let me blow this up a little. This colony is smooth. So is that one. So is that one. This colony is rough. And so is that one. I don't see any other rough colonies specifically. Maybe that one there. But students have a hard time looking and determining that. And you just have to learn by experience, I guess. One other colony characteristic is whether their appearance is glistening or shiny or dull. I blow this up again. So that one is shiny. That one is shiny. That one's really shiny. That one is dull. That one is dull. Students have a little trouble with that, but not too bad. So, and then the last characteristic, actually two characteristics to discuss. One of them is the pigmentation of the colony. If the colony has any color, it is pigmented. If it is not pigmented or non-colored, we call it non-pigmented. So this one is yellowish. This one is also yellowish. So these are all pigmented colonies. This one here, that one there are not colored. This one was white, that one's white. That one is off-white or opaque or gray. That one's kind of grayish. Anything with no color, and that's usually off-white, white, or I don't know, beige or something like that. We call non-pigmented. I'm not sure about a black colony, if that's considered pigmented or not. For this case, we'll call it pigmented, although I'm not sure black is a color, so I could be wrong on that one. Because usually pigmented means it has a color. And I don't see black in there, so I'm not sure. But usually colonies are not black. And that's a mold colony, which you don't want growing on your petri dishes, by the way. And then the last colony characteristic is the optical property. Most colonies are opaque. A few are translucent, and then very few are transparent. I think all of these colonies are opaque, meaning you cannot see through them. I've got at least one in here that would be translucent. only seeing it here, but I know there's another one there, right there. This larger colony is translucent. You can see partly through it. You cannot see there. On this one here, this large one in the center, it's opaque, but on the edge, it's translucent. So most colonies are opaque, like this one here, that one there, but a few are translucent, and then only a very tiny amount are transparent. Transparent colonies would look like a drop of water on the dish. And like I said, they're very rare. Okay, so those are the different colony characteristics, and this shows you colony A is a large colony, pale yellow in its color. That's very lightly colored. Circular in its shape. You could also call that irregular in its shape. Shiny, bulvinate, that I can't see from the photograph, and the edge is entire. Colony C, small colony. I would have to look at that non-blown up. I'm not sure that's really small, but we'll call it small. White in its color, circular in its shape, smooth, convex, entire margin. When we're talking about colonies, it's important to realize that we have two terms, colony abundance and colony diversity. Colony abundance is simply the total number of colonies on an agar plate. That's too many for me to count. So let me come up here. And if we say the number of colonies right here, the colony abundance is simply 4. Even this is a little too many to count. That would be something like 30 to 50 colonies. I'm not going to count them. And it'd be hard to count all of these right there because they're mixed together. But something like 30 to 50 colonies would be the colony abundance. So it's just the total number. Colony diversity is the number of different kinds of colonies. You have to go on the plate. That's a bit too much for me to distinguish, so let's come up to this one here. And then decide whether the colony is the same or not. Like all of these colonies and these big ones here too are probably the same species because they have the same colony characteristics. These are a little bigger than those, but these are more isolated than these. And that's probably why these are bigger. So they have more resources available for them to grow. They may also be bigger because this one in particular was started by more than one cell. And these were probably started by one cell. So that could be explaining why that one's bigger than those. But my point is, all of these are probably the same species. So that would be a diversity of 1. That would be a diversity of 2 because that's obviously a different species. That's a different species. That's a different species. That's a different species. So how many do we have here? 1, 2, 3, 4, 5, 6 different species. So we have at least 6 different species on this plate. So the diversity is something like 6, maybe to 8. Determining the exact diversity, it can be, I don't know, confusing because you don't know, is that just a small one of that? Sometimes it's hard to distinguish. And are those two the same or are they slightly different? So the abundance, you just simply need to count all the colonies. And if you come up with a close number, you'll get it right. If you're way off, though, it'll be wrong. And then the diversity, like I said, as long as you get a number that's somewhat close, I'll give you credit. But if it's way off, I will mark it down. I guess I should mention this when I'm grading your lab assignments. You should come up with a good answer. So think about your answers. Try and get a correct answer because I actually read all of your labs and all of your answers. If you're used to a instructor, particularly a lab instructor who never read your assignment and then you simply wrote something and it was graded correct, I don't do that. I will read your answers and it has to be a correct answer to get full credit. So let me warn you, you should give a good answer and think about your answers. All right, in this image, what's the abundance of wild cats in this photograph? So
7.
please give me an answer. That's correct. And what is the diversity of cat species?
2.
Yes, there's two cat species, the cheetahs and the lion. Five cheetahs and two lions for a total abundance of seven. Good job. All right, so you should know the terms in this lab. There's a neat picture of somebody's hand. You don't need to go through the references, but I skipped. Let's go to the worksheet. You're not actually going to be doing this lab because everything we do is online. However, you are expected to understand the concepts of this lab, so read through the lab module and then come to the worksheet. And in procedure one of the worksheet, we're going to ask you what is in the air you breathe. We have a plate that has been exposed to the air for doesn't say for how long, probably 20 minutes. And then it was incubated for 24 to 48 hours in an incubator at 30 degrees. And what's the colony abundance on this plate? And what's the colony diversity on this plate? You just have to look at the different colony characteristics to determine the diversity. Like that colony is clearly different than that because of different colony color. These two may be the same colony, they could be different, but that's clearly different. Those two are probably the same colony because they have the same color, most of the same colony characteristics. That will be a different colony because it has a different margin. And that could be the same color as that one, and maybe that one. Here will be a different one too. Anyways, you have to figure out how many different species you have growing on this plate to give the diversity, and then they just count the total colonies for the colony abundance. And that's question procedure 1A and 1B. And then procedure 1C, what step would you take to reduce airborne contamination of a sterile agar plate while you are inoculating it? So here we have a plate. Pretend this is a plate. We take off the lid. I'm going to put the lid down because it's too difficult for me to demonstrate how to do this holding on to the both the bottom of the plate, which normally I'd have that the on the table because I have to hold it up so you can see it. So I'm going to
We
put
can't actually see you, just your background.
you can't see it now?
We cannot see you at all, just your background. It's been, yeah, last lab and lecture it was like that too.
Oh, really? You're supposed to be
Yeah.
able to see me. Hmm. Um, Usually if I put it before my face, you could see it, but you can't even see my face when I'm showing
Nope, just the welcome week background.
all right, let me go like that, and then I'll maybe get it to work. Let me stop the share, and hopefully you'll be able to see what the heck I'm doing. All right, can you see
Nope, just background still.
Yeah.
I'm stopped the share. You can still see the background?
Yep,
You have to try to
just
start
your
the video.
background. It's like you're Your camera part is off, but your background is there.
No, the video is on. I'm not sure
Mm.
why that's not. When I had the share, you have to see my screen, and you can see the background when I have the share on.
Y-
Shouldn't
just
see,
up in the little picture thing of who's taking part.
so you don't see me.
Mm-mm.
Yeah, this has changed the way this is working.
Yeah, I can search for it. And also for the 6:30 lab, we didn't see you either because I go
Okay.
to the
No,
6:30.
I'm sure it's working the same. It's just changing the way they have it. So there's no way for me to show what the heck
So
I'm
I'm gonna
trying to
do
show you.
something.
What?
I'm in my car right now, but if I turn on my video, you see my background, but it's like now your screen's black all the way.
I'm supposed to be sharing, and this is supposed to be me. What happens there? This is smaller picture, and that's even smaller. So I'm not sure why that's not sharing me. See if that goes off. Still not seeing me. It doesn't share sound, but that's okay. Now I'm not sharing my screen, and all you see is the background. Let me see if I got this. Up here, but let me. Screw this up. Let me see how to get this back. There we go. Yep, it's not showing myself. Oh gosh, that's totally down now when I have no background. That's not very good either. I don't know what's going on here. I'm assuming you can't see me. To me, it's just a black background.
Just a black screen.
Yeah, I'm not sure what's going on, and I'm not sure what to do. All right, I'm not able to show you, so I'll try and hopefully you can see my screen now. Can you see my computer screen?
Yes, we could see it.
Okay, great. Um, so I'm trying to illustrate. Oh, what steps would you take to reduce airborne contamination? So pretend we have this petri dish right here, and I take off the lid, and then pretend my mouse is a loop, and then I'm working, streaking the plate with my loop, and then I put back the lid. What steps could you take to reduce airborne contamination while you are inoculating the plate? So don't tell me what steps you can take after you put the lid on, then you put the plate in the incubator. Don't tell me what steps you do before you're inoculating the plate. Tell me what you can do to reduce airborne contamination while you have the plate open and you're inoculating it. Procedure 2. What gets on your arms when you put your arms on the lab bench? So we have swabbed a lab bench And this would be the swab of the lab bench before you sterilize the lab bench. And this would be after you swab the lab bench with, well, you've decontaminated the lab bench. You don't really sterilize it, but you put disinfectant and clean the lab bench. And procedure 2A, did table disinfection work? That's just simply yes or no answer. And then what is the purpose of allowing the table disinfectant to air dry? Or once you put the disinfectant on the table and then spread it around, why do we allow the disinfectant on the table to air dry? Why do we not wipe it totally off right away? Any questions about that? So think about this answer. This one's a little tricky, a little more difficult.
I have a question about procedure 1.
Uh-huh.
The little dots surrounding the other colonies, do you count each one of those as well
Talking
for the
about this one and
abundance?
that one?
Yeah.
Yeah, those are colonies.
Okay. Thank you.
All right. Procedure 3. What is on your hands? What we did was we had somebody touch a plate with their fingers. There's 1, 2, 3, 4 fingers, right hand and the left hand. So there's the palm actually there, and there's the fingers this way on this one. And then they go wash their hands. You have to dry your hands because you can't touch the plate with dry hands and then touch the plate with wet hands because the water will wick the microorganisms from your plate, from your hands to the plate. So you have to dry your hands and then touch the plate after you wash your hands. And this would be the plate before washing your hands, A, and then the plate after you wash your hands and then dry your hands. And let me warn you that if you were to wash your hands and then touch the plate with wet hands, you will have more bacteria on your washed hands because the water wicks the bacteria from your skin to the plate. And I learned that actually in my first year of teaching that students were getting more on their clean hands, and it's because they weren't drying their hands. You need to dry your hands. Otherwise, you could, I guess, wet your hands before you wash them and then touch it after you wash them. As long as they're both wet and the same degree of wetness, it would work. But it's easiest to have them both dry hands. And there we have dry hands. Question 1, does hand washing work? And then question 2, suggest one reason why hand washing did not result in the complete removal of microbes. You may need to go online to get an answer to that. It has something to do with the normal microbiota on your skin. It has something to do with the hand soap that we're using. For example, if we wash our hands with regular hand soap, you'd see something like that. But if we were to wash our hands with regular hand soap and then put them in disinfectant bleach, washing them with bleach, we'd have less bacteria. And that would be an answer for 2. Why did you have less bacteria when you put your hands in bleach than when you simply wash your hands? So I'm giving you a little hint there. All right, procedure 4. When you're kissing your significant other, exactly what are you giving him or her? What's on your lips and what's on your tongue? So here we have somebody who touched their lips to the plate, the bottom lip and the top lip, and this is where they touch their tongue to the plate. Please note that I've given you a note above the picture that you should read about that picture. And then which plate has higher colony abundance? The lips or the tongue? And then on procedure, is that 4B? Why do you think this is the case, even though you might expect the abundance of both body sites to be the same, considering they're next to each other, and you often probably lick your lips with your tongue. So why is there an observed difference in the colony abundance between the lips and the tongue? I'm going to warn you, you need to give 2 reasons why one plate has more colonies on it than the other. And they must be correct answers. There's 3 easy correct answers. And actually, students have come up with a total of 5 different correct answers. But you must give at least 2 answers for why there's more colonies on one plate than the other, or on one plate than the other. Any question about that? And then C, why was the tongue plate incubated at a higher temperature than the lip plate? And I didn't go through that, but it says the lip plate was incubated at 30 degrees C, and the tongue plate was incubated at 37 degrees C. Why did we grow them at different temperatures? Procedure 5, microorganisms in the soil. What we did was we transferred a little soil into a tube and then of water, and then took a bit of that water, and then like 1/10 of a mil, and then spread it over this plate, and then allowed it to incubate at 30 degrees for 24 to 48 hours. And what is the colony abundance on this plate? Is it even possible to accurately answer this question? And please explain your answer, meaning what the colony abundance is, and explain why it's not exactly possible to answer that question. And with procedure 5B, answer, is there greater abundance of colonies from the soil plate to the lip plate? Okay. And yes, the lip plate has mostly small colonies. On the soil plate, let me blow this up a little bit. There are some colonies which are very large. These right here, for example, but there are a number that are very small, like those right there. And right here, you can't distinguish the colonies, but there are some very small colonies. So don't say simply because the lip plate has smaller colonies that there are more, because the soil plate does have some small colonies as well. So answer is the greater abundance of the soil, the lip plates. And then that's it. Go ahead and start working on the lab.